SylviaSpengler. I'm in the program in Mathematics and Molecular Biology at UC Berkeley and also in the Human Genome Center at Lawrence Berkeley Laboratory. I study Kinetoplast DNA, which is an array of some 7,000 molecules, circles of DNA. It comes in two sizes. A very large circle, there are about 10 of those, and all the rest are smaller circles, about 5 to 10% the size of a large circle. This array can be made planar, so that it's basically a tiling pattern that contains both the genetic material and also apparently some structural material. The actual role of the small circles isn't known, but what we're initially trying to discover is how they're arrayed. If you take the very large complex and break it apart by some sort of physical means, you can get subunits. Subunits vary in size all the way from the single small circle to arrays of up to 10 molecules that you can actually analyze. What we first look at is what the linkage is between two circles. And what we have shown by looking at some 600 molecules that have only two circles in them is that it's a singly linked catenane. It's the simplest sort of catenane. However, we have yet to answer the question of what is the number of neighbors that each single circle has. We think it's probably in the range of 3 to 4, but even that doesn't tell us exactly what the array looks like. For example, you could have it be 3, and when you separate it by gel electrophoresis, you would be able to separate something that was 3 in a linear array from something that was 3 as a triangle. Even when we have determined how all of these circles are linked together, one of the big questions will remain, how does it replicate? What happens is this entire array doubles its size. And it is done by each of these circles coming off the array, duplicating itself through a knotted intermediate that we still don't know why it's a knot, recircularizes and reattaches to the entire array to make something that is twice the size of the original array, which is then split down the middle. It happens very, very rapidly. Biologically, the question is how it happens and how it could be arrayed so that it happens rapidly enough to let the cell survive.